student. I hope you understood the previous question that we have learned for the NEET paper from paper code R1. We are going to solve the next question. So I'm going to share my screen and let's solve that. By the way, I hope my screen is visible to you. Even if it is not, it's okay. Till the time I'm speaking, it will be visible. So I'm repeating this. This question that you're watching, this paper code is R1, and this is NEET 2022. This paper that happened in July 17. Yeah, July 17. Anyways, now we are doing the second question. I repeat myself, section A, which is botany section. 101 question has been done, and this is question number 102. If you see this is asking question from gel electrophoresis. That means question has been asked from the chapter biotechnology. Yeah, that chapter is very much important. You should do it in a proper. So now let's solve this question. Of the following statement, this is not true. Guys, you know what? There is a maximum tendency in the kids that they do not learn the statement or read the statement properly. And there is then chance that they can make mistake. Like the last question we solved, they are also not true or not something like that. So that's where you have to understand what exactly it is asking. Anyways, question is regarding gel electrophoresis. That four statement has been given and you have to find out which one of the following statement is wrong because not true, and three statements are actually right. So let's identify what these statements are. The first question is bright orange color bands of DNA, it can be observed in gel when exposed to UV light. I hope you do know that UV light is used. By the way, if I tell you the short story, DNA normally it has negative charge, right? Because of all the oxygens, lone pair and everything, if you remember phosphate group, and it is attracted toward the positive charge during gel electrophoresis. I'm writing this. This is attracted towards, yes, my handwriting is not very beautiful, so you can put it in the comment if you don't get it, okay? Attracted toward positive charge. Anyways, that's the reason that when we are, separating DNA molecule, we reacted with restriction enzyme, because restriction enzyme will cut the DNA into different segments, and then all these segments are moving by the differences in the molecular weight. Because we are using agarose gel, I'm just putting a reminder there, we are using agarose gel here, and into the agarose gel, because you know that there are pores present inside, because it is a gel made up of the agar. Agar is extracted from red algae, Gracilaria gelidium. I'm telling you so much right now, but yes, DNA, which is having high molecular weight, cannot move up to, like, suppose this is gel. So, and there you have put the DNA into the small cavities, or we call them, cuvettes. Okay, small wells also. So DNA, which is having high molecular weight, it cannot move up to a larger distance. So it will stop at a smaller distance. DNA, which is having a smaller fragment, small molecular weight, it can move up to a larger distance. And that's how we separate DNA from gel electrophoresis. Yes, after the DNA separation has been done, all the DNA has moved to the places, then we do visualize the DNA, and we see the DNA with the help of ultraviolet light. And please remember that, because it visualizes the DNA. DNA actually is not very colorful and everything. We have to make it colored. It is almost transparent. How do we make them color? Let's read more statement for that. Okay. So the process of extraction of separated DNA strands from gel, this is known as elution. Elution, this is a term which is used for separation. If you do remember learning chromatography in photosynthesis chapter in 11th class also, elution term is used, that how substance are being separated in the chromatography. So elution is something which is used for separation, not only in chromatography, otherwise also. This term is true because we are separating DNA, right? First term is also right, because I told you, UV, first section or first line you may say. Third is the separated DNA fragment are stained by using ethidium bromide. Okay, let's put it on hold and let's see fourth option. The presence of chromogenic substrate gives blue colored DNA bands on the gel. First of all, guys, I told you that a colored substance is used because we need to visualize DNA under UV light. So obviously we will be using a colored substance, and we are going to use a dye. I hope you do know or remember the definition of chromogenic substrate. Chromogenic substrate is a substance which like, if imagine I am a scientist and I'm using a dye, I will always prefer a dye which is not interfering with the main substance. I want to see the DNA. So the dye that I will be using, it should be inert. It should not interact or interrupt or interfere with the DNA structure. It should not do any kind of manipulation, right? So that is the reason that we prefer ethidium bromide, because ethidium bromide actually increases fluorescence, because we will be watching the DNA under UV light. So ETBR, that's the short form, that's how we say them. Ethidium bromide, it actually enhances fluorescence. It enhances fluorescence when we want to visualize DNA, and it is going to bind with, it's known as intercalating agent. That's the term, I think. You can cross check in chemistry, that was the term. Some kind of agent which will bind and just, enhances the fluorescence. And yeah, talking about the fourth point, why did I mention chromogenic substance? Chromogenic substrate is actually a substance which is a colored substance, but it interferes. Because imagine we want to study proteins, we want to study enzymes, we want to study the rate of reaction of the enzyme, then we use this dye, because the color of these dye changes as soon as it is reacting, as soon as enzyme is reacting, its color starts changing. And into the experiment, actually, we observe how much color change has been happened. So chromogenic substrate is also colored substance, but it is interfering. So it has a different, mechanism to work. And we do not use it in the DNA especially, because we just used it in the enzymatic experiment. You can read about it. Okay. So that is it. That makes fourth option as not true, because chromogenic substrate has totally different meaning, which is used in enzymatic reaction. Thank you so much. We are done with this question, but stay connected.